Wait. Okay, so we are running. Welcome to everybody. This will be our last webinar before the summer break for this year 2026. I'm very happy to have here Professor Bunyam Tar'an. He is a professor at University of Saskatchewan and is involved with the Ministry of Agriculture in mainly working on chickpea and flax. Today we're going to talk about chickpea and resistance to Canadian extremes. So Bunyam, please, the microphone is yours, and we'll have a discussion after.
Okay. Well, thank you very much, Nelson, for organizing this webinar. And I'm pleased here for the opportunity to share what we are doing here at the University of Saskatchewan. So my name is Bunyam Tar'an, and I'm a plant breeder. I'm working with two crops, chickpea and flax here for developing the varieties for Canadian environment. So today I will be talking mainly about chickpeas and about abiotic stress tolerance, which is the two extreme in Canada is cold and heat stress, which is now many of the, as I heard from Nelson, many of the European countries, the southern part, experiencing the high temperatures while the crops are growing. So this is a very timely kind of discussion what we will have today. So let me share my slides. Okay, so I didn't see it. Okay. Can you see my slide here?
Yes, perfectly.
Okay, thank you. So this work is done by a number of people. So I would like to acknowledge the research associates, students, and research officers. So Sweta, a former postdoc with our group, working on the cold tolerance, and Sophie is current PhD students with my group. Megan House is a research officer, and Kishore working actually with Tom Workington Group, research officer, working on helping us with some bioinformatics with this research. So here is the Canadian where the agriculture. So we are located right in the center of Canada in Saskatchewan. Just to let you know that about half of the agriculture in Canada is in Saskatchewan. So 46% of the annual crop production, cropping areas in Canada is in Saskatchewan. So it's huge agriculture in Saskatchewan. The total area of crop in Saskatchewan is about 14.5 million hectares. So, and this is where the chickpea is growing in the southwestern of the Saskatchewan and southeastern of the Alberta. That's where kind of the drier areas of the province where the chickpea is growing. So this is some statistic where we are now with the chickpea seeded areas in hectares in the last 4 years and yield and also the total production that we have. So in 2022, it's only less than 100,000 hectares were grown on chickpeas. Chickpea, by the way, is one of the latest crop added to the Saskatchewan or the Canadian agriculture. The commercial production is only started in 1997. So this is where we are now. So last year, 2025, the Canadian producer grew about 219,000 hectares of chickpeas with the yield on average about 2 tons per hectare. So you can see the yield variability. So 2022, 2023 was a little bit on the lower side because of the very dry conditions during that time. So latest production from 2025 is close to 500,000 metric tons. And 95% of the chickpeas grown in this region in Western Canada is the varieties developed at the Crop Development Center. So this is just to show you how the chickpea feel at the farmer level in Saskatchewan. You can see kind of seeded crop, full seeded crop, and a little bit gentle rolling areas where chickpea are grown in the province of Saskatchewan. So view like this, when you see like an open flat, open space, no trees, prairies, Saskatchewan, that's kind of typical farming here in Canada. So one of the challenges of the chickpea, especially the production in Canada or in Saskatchewan, is very short growing season. So this is probably the most northern hemisphere where chickpeas is being produced commercially. So usually we start seeding during mid or early May, and then we harvest mid to late September, and sometimes going into early October. So this is the typical growing period in Saskatchewan. As you see, temperature is increasing from seeding. The average temperature during the seeding time is about 10. So when the soil temperature 10, 15 degrees, then we start seeding, and then it's kind of trending, cooling again at the end of the growing season. So by October, November, usually we already have some snow, and then the snow stay on the ground until April. So the total precipitation in this region is quite low, so about 366 millimeter per year, the total precipitation. But 5, 6 months in a year, the ground is frozen. So with that, then when you look at the overall how the growing period, so we have two major risks for the abiotic stress. At the beginning, we may have a risk of freezing. So when the plants, the seedling growing, sometimes the temperature drop and become like frozen in the field. And also at the end of the growing season, sometimes when the pods is not mature enough, still watery, so we may have low temperature again and freeze everything in the field. Or sometime in Saskatchewan, we know what we call a killing frost. They usually happen on the 3rd week of September. And then during the growing season, in June and July months, we may have also high temperature picking up for a few days above 30, 34 degrees Celsius is often happen. So that's happening usually during the flowering. So that also having really affecting the yield. As you see at the beginning, beside dry, high temperatures affecting the crop, so that lower yielding overall in the chickpea production here. So this is the two stresses that we will be talking in this seminar. Okay, so low temperature is an important for chickpeas because it's seeded early in the spring in Canada. Night temperature could drop to below 0 and is causing freezing injuries in chickpeas. So then we start to look into, okay, is there any how to kind of this mechanism? Is there any tolerance in the germplasm that we have in chickpea, and then can we tap into that germplasm for our breeding purpose for improvement of low temperature tolerance? So with that, then we look into some germplasm. ICARDA also has some nursery, and we tested, but it's not strong enough for our environment. So then we tap into the wild species. So this is some kind of general why we use the wild is increase overall diversity, becoming a building block for our breeding program, and allow us to tap in for climate change adaptation and potentially to avert disaster because of the extreme conditions. And overall is helping for global food security. So the materials that we are using here is the kind of newer collection that were done about decades ago by the group consortium from many universities led by Dr. Doug Cook from UC Davis with collaboration from scientists from Turkey. So the group had collected over 2,600 new accessions of wild Cicer, Cicer reticulatum, and some Cicer acanthus ferment. And from those 2,600, they did some sequencing and then narrow down to 20, the small number that kind of captured the diversity that is the most diverse reticulatum and is distributed among the members for use in a breeding program. So the overall story about this new collection and their characterization was published in Nature Communication many years ago in 2018. So from the 20 that we receive from this collaborative research, then we just kind of doing the simple way. Is there any cold tolerance within this population? So basically here what we did back in 2017, 2018 with our former research associate, Dr. Donna Lindsay. So basically we seeded late in the field. We seeded around late or mid-August in 2017, 2018, and then let the snow cover the plants, and then we clean up, and then we look at is there anything that look like promising from this initial trials. So from this we found there are about 5 to 7 that are look promising. So as listed in the bottom here. So QDA 152, Casson 075, Cicer 063, Oyali 064, 084, and Barry 3106. Those are among that seem to be having like promising for sources for cold tolerance in chickpeas. So then we did further characterization under control conditions. So this is the model that we are imposing to those materials. So basically we started with normal condition with 14 hours, 22 degrees Celsius, 10 hours, 16 degrees Celsius, about 2 weeks until about 5 to 6 node states. This is what we call a control. And then we gradually lower the temperature to 4 degrees Celsius for 1 week. And this is what we call a cold acclimation. And then we drop to -6 degrees Celsius for 24 hours. This is like freezing stress that we're imposing. And then after that, we put back under normal condition for up to 3 weeks as a recovery. And then we do the scoring after the plants came back into the recovery. So with this, and we can see clearly, so we just use the simple scoring that was published many years ago by Singh in 1989. So basically just cold tolerance rating from 1 to 9, 1 meaning there is less symptom of damage to the plants, and then 9 plant completely dead. So you can see that here on the left one, QD 152, Casson 1075, and 2 other wild species, which is showing like a more tolerant, less damage. Whereas on the cultivated species, CDC Consul and CDC Leader on the right, on the picture, on the graph here, after 2 weeks and 3 weeks putting back under normal, you see there's no recovery and plants are completely dead, basically. Excuse me. So you can see on the picture on the right here, for example, Casson 075, we put 4 nights at -5 degrees Celsius. Compared to Consul, just 1 night, you see Consul start really damage from this low temperature, whereas Casson showing basically normal, very small damage to the plants. So, and then we look at the timing, okay, how long the plants can withstand these low temperatures. So we expose them to -6 degrees Celsius for 3 hours, 6 hours, 12 hours, 18 and 24 hours at -6 degrees Celsius. And then after exposing to -6 degrees Celsius for that given time, we put them back into the normal chamber. And then after 1 week, we did the scoring. Okay, as you see from this graph here, the 2 wild species, QDA 152, after 18 hours and 24 hours, the whole time frame here, still showing some recovery and plants back to normal, whereas at the bottom, CDC Consul and Leader, which are the current cultivated varieties, completely damaged. There is no recovery. So seems to be the limit here about 18 hours. So by 12 hours, the cultivated species still be able to recover. But when you pass 12 hours at -6°C, there is no recovery of the cultivated chickpea species, whereas the wild species, the two sample here, QD, which is collected from Mount QD area in Turkey, and Kasen 075 from Kasentas near Diyarbakir, Turkey. So those are the ones that we use in research here. And then we look back here, and what we did is that we are interested to do RNA sequencing before and after freezing. So we look at, this is the time point where we collected the tissue sample of the control plants and plant exposed to cold temperature. So we started from control, cold acclimation, and freezing at 3, 6, 12, and 24 hours. And then recovery 1 week after we putting back into normal conditions. So we used two samples here, the Kasen 075, the wild, and the chickpea variety CDC Consul for this analysis. So this is the data, the Venn diagram showing different kind of comparison, 3 hours versus cold acclimation versus control for both samples that we did, CDC Consul and Kasen 75. So what happened is that the majority of the genes expressed compared to control is during the acclimation. That's when the plant's kind of responding to the signal, to the lower temperature stress, and then kind of we start looking at, okay, what kind of reaction that happening within the two plant samples here. So you can see the number here, recovery versus control, 24 hours versus control, 12 hours, and etc. So there are a lot more analyses were done. So basically, when we summarize all the genes that involve in this regulation, there are kind of two groups we can put together. So the upregulated is about cellular response to stress, cell death, ethylene biosynthetic process, jasmonic acid biosynthesis, salicylic acid biosynthesis, cellular response to abscisic acid stimulus, response to hormone, respiratory burst, response to temperature stimulus, and then downregulated about anatomical structure, morphogenesis, cell cycles, phase transition, cell proliferation, chlorophyll biosynthesis, cytokinesis, developmental, photosystem, and regulation. So these are the two main groups. And after putting all the data together, basically we kind of follow the model where basically involved in the calcium influx and map kinase that mostly involved. So by looking at, for example, here, this is kind of the hypothetical, the model, where you can see one of the central role here is the C-repeat binding factor, CBF, play critical roles in this analysis. You can see on the right here the heat map, the expression. Okay, so the column on the right here is kind of alternating between Consul and Kasen. So you can see if there's any differences between the two groups. You will see in this graph here. Okay, and then also we look at the overall, so calcium dependent kinases, CIPK, CBL, CPK, and MAP kinases. So these are the ones that all affected by in response to the low temperature between the two samples that we have here. Okay, and again, there are also a number of key transcription factors that are that are active in response to the low temperature, and where we can see the response between the two samples under different time points that we collected. Okay, so all of this are really pointing out to the data cycles or the model that we saw before. So Again, this is some example where the expression pattern of common differentially expressed genes in at least 10 treatments between Casson and CDC Consul. Okay, number of highlight here, including gibberellin, number of transcription factor, and as well as some gene related to the photosynthesis. So this is what we hypothetical mechanism of cold tolerance in chickpeas. Basically, the cold stress, there's a perception through the plasma membrane and going through calcium signaling, and there's a phosphorylation cascade with the active MEP kinases. And these are all the transcription factors that are kind of upregulated in Casson. And then there's cold response gene like IC1, CBF2, CORE, LTA, and MIP15, which is the negative regulator, and then also interaction with the hormone gibberellic acid and abscisic acid that leading to the cold tolerance in these samples of chickpeas. And then we also did QTL sequencing. We created F2 population by crossing those two lines, Casson 075, which is reticulatum, and CDC Consul. We cross, and then we created an F2 population, and then we exposed the F2 population to low temperature to -6 degrees Celsius. And then we collected the bulk DNA from the tolerance and sensitive, and we did the bulk sequencing on the samples here. So basically just following the typical protocols with SNP index. And then what we found is that the QTL that we identify using the bulk sequencing is kind of overlap with some regulators of the cold tolerance mechanisms such as CBF2, a number of transcription factors. So when you look closely in the regions where we identify the QTL, a strong QTL, strong index here, there are also transcription factor AP2, MIP, WORKY, BAS, LAH, NAG, as well as GA20 oxidase, all kind of within some of the QTL that identify using this QTL sequencing. So all exhibited a significant differential expression between Casson and Consul, which are the two parents of these populations. So from here, kind of the takeaway from the cold here is that the higher freezing tolerance were obtained in two reticulatum, Casson and QD152. RNA sequence revealed genes related to stress response, endogenous stimulus response, secondary metabolite process, photosynthesis were differentially expressed between the two lines. And then the expression of CBF pathway-related genes play a critical role in freezing tolerance in Casson 075. And QTL sequencing identify genomic region associated with freezing tolerance, and an F2 population, which is kind of overlapping with the RNA sequence data. So there are about 58 differentially expressed genes that co-located overlapping with the QTLs that were identified by this QTL sequencing approach. So now we switch the gear into the heat stress. So this is happen usually in June and July. Usually that's happening when the chickpea in the field in Saskatchewan is during the peak flowering or start the podding stage. So this is just an example here, okay, the 2021 North American heat wave happening, and it's more often is now frequency, more like a higher frequency may happen in Saskatchewan. So heat stress in chickpea, really one of the heat stress and drought, one of the most common largest stressor in chickpea. Besides that, in addition to that low temperature, and the chickpea especially sensitive at 35 degrees Celsius, and it's not understand well the genetic mechanism. Oh, by the way, this I give this presentation as well during the ILS about 3 weeks ago in Dubrovnik, beautiful Dubrovnik, and thanks to the organizing committee for an excellent meeting. Everybody, we really enjoy the conference, the venue, and the location. So my apology if you were there, and then so this is kind of a repeated story I presented during that conference. So plants usually try to kind of coping with this high temperature by escaping, by flowering and maturity timing adjusting, by avoidance, for example, stomata response, leaf arrangements, and there's a so-called thermotolerance when the heat cannot be avoided. So this objective, this is part of the one of the co-authors of Dusain PhD program. So to screen a population to identify lines that have stable reproduction under heat, to explore the critical stage and physiology of its stress response, to identify potential candidate genes through GWAS and the integrating these genes into the yield lines. So this is the same material that we use initially from the collection of new reticulatum. So we used 19 of the Cicer reticulatum and we crossed with CDC leader. And then in the F2 of these two stream of crosses, we intercross the F2. So that create kind of really high variability. And from this, we selected initially over 2,000 lines and then narrow down to 600 lines, the one that can survive under Saskatchewan conditions. And then by F5 and continue to F8, we have 200 lines initially selected for seed quality and resistant to Ascochyta, one of the major requirements to be able to grow in our environment here. So we genotype here using our own Axiom SNP chip, CDC Cicer, 60,000 SNP chip. And these are the diversity neighbor joining tree based on 35,400 SNPs, showing the genetic relationships among these 200 interspecific inbred lines samples that we are using. So initially, we grow these lines under 2 seeding dates in Saskatchewan, 2 locations in 2019 to 2020, and also in Yuma, Arizona, in the warm desert areas in the United States. So this is kind of the maximum temperature at flowering at seeding date 1, which is normal seeding date, and seeding date 2, which is late seeding date. So on average, you see the maximum temperature reached during the flowering. At normal, it's about 29 in Yuma and 26 or 27 in Lucky Lake, Saskatchewan, whereas under seeding day 2 in Saskatchewan reaching 35.5 degrees Celsius, whereas in Yuma on average reaching about 38.9 degrees Celsius. So we measure we collected all the agronomic data and yield data, and then calculated some index related to tolerance, mean productivity, abiotic tolerance index, and stress susceptibility percentage index, modified stress tolerance index. So all these index we calculated based on this 2 performance. And this is just an interspecific population nursery in Yuma, Arizona in March 2020. So we all still remember 2020 was very challenging because of the COVID. So we were not able to inspect ourselves, but our collaborator back then in Yuma, Arizona, were able to manage the trial until harvest, and then sending us all the data that were able that they were able to collect during the trials. So from that, we did some GWAS and identify some QTL, as well as we did some preliminary prediction models. Can we predict the performance under stress or seeding day 2 compared to seeding day 1 as the training populations? And then we measure the correlations between the prediction accuracy, the correlation under using the prediction model as well as the actual. So you can see here I give some example here for Ascochyta. This is scores at flowering as well as at podding. And again, the trial were not inoculated, so this is just based on natural inoculations. So the prediction model for Ascochyta is actually kind of low to moderate. At flowering, about 22 to 28%. At podding, about 35 to 38%. Seed weight is the highest that we can see the prediction between under normal and under more or less like stress conditions. 0.6 to 0.7 and yield about 0.4, 0.5 to 0.6. So there's some potential there. And again, this is the same population, the same genetic materials, just under 2 different conditions. I would expect to be much higher than this, but this is what we have because of the variability. And also we did genetic analysis. So we evaluated these 200 lines under control conditions and collecting all the symptoms that were appear on the plants. So this is the model when we did the heat stress analysis. So basically we synchronized the flowering. So we collected the flowering date first under normal conditions, and then we adjusted the sowing date, okay, so that all the plants will have the same flowering, the same state of flowering when we expose them to high temperature. So the first buds, and we gradually increase the temperature for 6 days from the 22 degrees Celsius all the way to 35 degrees Celsius during the day, and then 29 degrees Celsius during the night for 2 weeks, and then gradually decrease the temperature to normal conditions. So this is done in 2 runs. In each run, we have 208 entries, 2 treatments, which is control and heat stress, and 3 replication in each run. So we have very strong data for this. And also we did some microscopy, physiological analysis and transcriptome analysis on this population, on selected lines on this, which is we change the temperature to be higher at 40 degrees Celsius during the day and 25 degrees Celsius during the night temperature. So basically, probably if we grow them in Italy or in southern Spain at the moment, maybe we don't have this chamber, because I believe the temperature there is quite high at the moment. So this is some of the data. So seed yield, for example, when we compare under heat stress, okay, how much we can get yield per sample, you can see the population is kind of maintaining quite high compared to the other 2 groups of parents. So on the left here, the red dot is on the elite cultivated lines. On the right, the blue dots are the wild species parents, and the green dots are the hybrid or the interspecific population. And then you can see the seed yield difference between heat and control. Okay, so the wild species still maintaining, but you can see the progeny from the crosses. Also, there's some lines that kind of maintaining at high yield under control and under heat stress. So this is what we observe. There are a number of symptoms you can see when plants are exposed to the high temperature. So actually, there's some increase in branching, but the branching is not that strong. So then the overall biomass there's a reduction, and there's a early maturity, and then this the overall can cause plant death. So on the plants level, you can see the leaf scorching, so changing in chlorophyll, anthocyanins, proline, oxidative response, lipid peroxidation, membrane damage, and relative water content. You see also leaf loss, can flower abortion, and probably due to reduced pollen viability, and there is pollen structure damage. And also we see some immature pods and empty pods from this heat stress effect. And then we did the GWAS results, so seed number under heat, harvest index, yield difference, 100 seed weight, so everything kind of line up nicely, and you can see some overlapping. And then interestingly, this the majority, the strongest QTL here is located on chromosome 4, which is overlapping with already known, well-known drought QTL hotspot in chickpeas. So there's also a number of other factor or candidate genes. One that is kind of really significant is a sugar transport protein, MST4 like gene within this population. And this is the effect of specific lines. So we pick up 3 lines, 76, 168 tolerance, and 147. And sensitive line when we expose them at podding, flowering, and at flowering at different temperatures. So 40 degrees Celsius, 25 at night at podding stage, and at flowering, you can see the the tolerant ones be able to withstand and produce some yield, whereas in elite checks there is no production, plants are dead. And then at 35 degrees Celsius, 29 at flowering, plant respond much differently from the 40-25. So actually, there are more damage on these tolerant lines at 35 degrees Celsius and 29 at flowering, whereas the elite checks slightly maintaining, although lower, but you can see not much kind of reduction under this 35. Okay, so this is the how plant responding. So this is kind of when the flower appears, so under the heat stress treatment. So from 22, 30, 40 degrees Celsius, and then going down to 30, we tag every single plant that appear in the given day, and then we follow how the development, and we compare the most sensitive and the most tolerant. So you can see here the most tolerant were able to continue to produce mature pods, continue to produce mature seeds, whereas most of the sensitive, all the sensitive that flower during this period, mostly aborted flower during that period. Okay, so what happened is that flower loss happened in this population. 10% happened pre-pollination stage and 90% happened at post-pollination stage. Okay, so this could be a number of potential mechanisms: pollen failure, pollen tube failure, fertilization failure, embryo development failure, or pod development failure. So with that, and we did some electromicroscopy, scanning electromicroscopy. So you can see when we compare the control and heat stress for a number of different lines, sensitive and tolerance. Okay, so the sensitive one, there is no viable flower at 40 degrees Celsius. And then in tolerance lines, you can still see some damage at 40 degrees Celsius. Okay, but the majority, the high percentage, the pollen still kind of intact. And if you look at closely, there's some kind of a rupture or kind of shrinkage, dehydration of the pollen grain under heat temperature here. So we're now looking at into the structure of this pollen. So probably this because of like a thicker exine or others, as well as you can see here. Okay, so the pollens and a lot of our aborted pollen grain within the sensitive compared to the tolerance lines on the left here. And also pollen tube growth under heat stress, the control continuing, whereas at the heat stress, the sensitive, there is no pollen tube growth compared to the tolerance samples that we have here. Okay, so we did also all the physiological measures such as formation, electrolyte leakage, relative water content, proline, anthocyanin, chlorophyll A, chlorophyll B, chlorophyll total, carotenoid, P2 and PN0. So then we did some correlation analysis. Some of them are really negatively correlated with the yield under heat stress, and some are positive correlations. So look like there are some potential different mechanism here. For example, proline content actually higher in the sensitive line than in the positive, but there's also much lower in the cultivated sensitive, whereas the anthocyanin quite high in one of the tolerant lines that's showing a kind of 3 to 4-fold higher than any other line. So this kind of really what we see, we repeated this few cycles, and then we still get consistent results. So there's a potentially different mechanism. So key takes away from here, there's overlapping QTL for drought tolerance and heat stress. There's a diverse heat tolerant responses and more sensitive at 35 degrees Celsius, 29 Celsius at night than at 40 than 25 at night. Pollen sensitivity is a major issue, but also halted flowering and podding. So basically, it's a complex mechanism for this. So with that, I'd like to thank you for your attention and the funding support from the University, Government of Saskatchewan, Saskatchewan Pulse Growers, and Sustainable Agriculture Partnership, the cluster program in Canada. Thank you for your attention, and I'll be happy to answer if you have any question.
Thanks, Bunyam. It was a very interesting presentation. You presented a lot of very on-point results. So let me see if there's Any question from the audience? You can just unmute your microphone and talk. I will now also check the chat. So, okay, so there's raised hand and Azim, please.
Yeah, very nice sir, presentation. I'm very glad to see you here, sir, because before this time I just read your papers in different journals, Bunyam, so I'm really happy to see you. Sir, I just have a little query about it. Your heat stress experiments in the field. So, how much irrigation you are given to your experiment? Are you giving irrigation in Canada or not? Please, first. Sir, please open your mic, sir. Sir,
yeah.
Sir, yes, please.
Yeah, my apology. Thank you, Azim, for the question. So, in Canada, there is no irrigation, but
Okay.
in Yuma, Arizona, the plots were irrigated. So it's very warm in Yuma, Arizona. It's irrigated. So we try to separate between drought and heat so that we maintain that under irrigation. But in Canada, there is no irrigation. So it's just based on. So that's why when we look into that, Azim, under Canadian condition, the seeding date too is kind of stress under quotation. Okay, so because this is, we cannot separate between the two. So this is just initial step to give an indication. Is there any kind of line that potentially can withstand high temperature during flowering?
Okay, very nice. Sir, one more thing is the water holding capacity of soil is also matter for heat stress tolerance. Is it, sir?
Yeah, we didn't do that. So probably the next time, okay, we will do that. But again, when we do under control conditions, when we kind of fine tuning the heat experiments, we maintain the water content in the media at around 60 to 70% fill capacity. So we continuously monitor by weighing, by the weight, and then adding water. So we kind of confident that the response under control conditions in the photosynthetic chambers was due to heat stress, because we continue monitoring the soil water content and then adding when it's needed.
Okay, sir, one more, please, if you don't mind. Sir,
No.
you did your experiment for heat stress in net house or glass house or in the field.
Can you repeat again?
Sorry, I said you did your experiments for heat stress in the field or in net house, where you did your experiment for heat stress.
So the first experiment, the initial screening, were in the field. So by adjusting the seeding dates. Okay, so we know how the pattern of the temperature. So we adjusted to normal condition, and then 2 weeks later we seeded the second experiment. Exactly the same number of replication side by side, but different seeding dates. So the idea is that to expose the plants at flowering to high temperatures. So 2 seeding dates at 3 locations, two in Saskatchewan, 1 in Arizona. That was the initial at agronomic level. And then the second round, when we did the experiment, is in the phytothorn chamber, growth chamber. We have big growth chamber that can hold up to 600 pots. And then we adjusted the temperature, kind of gradually increase temperature to 35 degrees Celsius and 29 degrees Celsius during the night from there. And then from there, kind of really fine tuning there. And then also we adjusted the sowing date so that all the plants will be exposed to high temperatures at the flowering stage. So that's the kind of the challenge. That's the challenging part, Azim, to synchronize the flowering, because otherwise, if you just see the sowing at the same time, then the early one will flower early, and then the late one will not be kind of at flowering when you expose to high temperature. So that was the challenging part. We basically we sow a lot, and then just pick up those that kind of start developing the buds when we expose them to high temperature.
Okay, very nice. Sir, one more, please. Sir, when you make your crosses of Cicer aratinum with Cicer reticulatum, the color, size, and quality of grain is affected there?
Well, when you make interspecific cross with the wild species, maybe you need many generation of selection or back crossing to bring back the quality. So that's a different story. As in, if you are doing the breeding, market will not like it. There is no markers but on nutritionally it's not bad actually. We did the nutrition analysis like protein content, iron content. Actually there are some good in this wild species, but visually when you look at the seed quality, it's really kind of almost like an inedible. People will not yeah.
Sir, actually, I did my crosses here of reticulatum with aratinum, and I have a success to develop a variety in which the blood of reticulatum is included. So by using the reticulatum, it's a good chance for us that we can develop our lines that are tolerant to freezing and also for heat tolerance, because when we use this reticulatum blood, it's also increase our flowering size. The duration of flowering is more, but the problem is only here its grain size are a little smaller than the aratinum. But when with many crosses, it will be going well. Thank you so much, sir, for having a discussion. Thank you.
Thank you for the questions.
There are other questions, the burning questions. So first one is Tom.
Thank you, Bunyam. Very nice. Can you tell us a little about how you will be planning to use the data arising from these kind of fundamental studies on freezing and heat, how you will bring these data into your breeding strategy?
Yeah, that's a very, very good question, Tom. So we try different models, Tom. So if we identify potential key genes that we are expecting, so then we introgress them using the accession, for example, cross into the cultivator. So we have the population now, and we look into the genes, but we may miss something there. So then we kind of now use the population to develop a genome selection from this. So kind of two ways. So targeted gene as well as the background to recover by genome selection. But again, even in the genome selection, our preliminary analysis, although they are the same lines, just different in seeding dates, we use that as a test population and training population. The correlation is not that high that I would expect. So probably we need to use different model by incorporating G by E, something like that. So this is kind of just very preliminary. And again, probably we need Nelson to help with the bioinformatics because we are kind of amateur, Nelson. We are not into that. So we have Cicer helping, but again, we are learning as we are doing this. So both ways, Tom. So we targeted if there is any strong gene, candidate genes, we use that in marker assist selection and introgressing, but also the background. We don't want to miss anything because this is a complex, complex trait.
Thanks. Thanks also for the kind words. Carlo.
Yes. Good morning. Good evening. Thank you. Excellent, fascinating topic. Tom sort of framed my question, but I want to follow up on his question by asking if a plan could be to use these QTLs that you have detected to screen the broader germplasm, Cicer germplasm collection, the cultivated, because I'm hard to believe that there's no better variation within the cultivated domain. You just need to find it, and this is a great tool to find it. Okay, see which accessions carry these markers, and at least you can try them. Okay, that's one of my first question suggestions. The other one is there a way, because we have a similar situation here in North Carolina, is there a way to using digital phenotyping in these contrasting lines to see if you can predict better the reaction from the phenotypic perspective? For example, canopy temperature for us in peas has yielded very good results in terms of tolerance to early heat and effects on flowering. You may argue that the tolerance is due to mainly anther characteristics, but sometimes it's the canopy itself that heat up too much and hence kills the pollen. Okay, so some ideas and some suggestions in terms of using this information more quickly in the breeding perspective, because if you find diversity within the cultivated domain, is a straight pathway than trying to introgress these genes from the wild. Okay, just thinking about, but it's great presentation.
Yeah. Thank you, Carlos. So we haven't screened the whole cultivated, but the Icarda formally has some cold tolerance nursery they have. So basically, they have selected among the best germplasm they have. Even before the war, they have that very active nursery. So we tested that under our condition here. It's not as strong as what we see and with this cultivated species. Okay, so there's a limitation there. So there are some variability. So for example, when you expose over 18 hours at -6 Celsius, many of them basically never recover of the germplasm. We have a so-called association mapping panel, which is kind of very diverse as well. And we tested that before we go into this, and we didn't see that. But it's kind of limited, about 300 lines diverse that we use. Maybe if, as you said, if we expand that into broader germplasm, we could find one that maybe similar or on par with what we have here. And the second comment you have about digital phenotyping camera, we haven't done yet. Probably that's the next one. Again, we don't have the expertise, Carlos, at the moment, so we just follow like conventional ways, looking at the response of the plants using the tools that we have. So that probably that's probably one of the future that we are trying to do, and then we can compare to what we have right now and how kind of precise it is. Thank you for the comments and for the suggestion.
Thank you for the presentation. Thank you.
Okay, I have two quick remarks at the end. I don't remember the exact nomenclature you did, but you thought that the response to heat could be divided like actual adaptation, like closing stomata or adapting the direction of the leaves, and then some more intrinsic resistance, like the plant is tough, is able to withstand it. Were you able to dissect the kind of stress and stress resistance that you've seen toward, okay, this is happening because of adaptation or resistance, or it was not part of the experimental design?
Oh, yeah, the answer is it's not part of the experimental design. So what we are looking for here is number one, is there any tolerance to high temperature? And then number two, what is the potential mechanisms? But we didn't see kind of the stomatal or leaf curling or anything like that during these experiments. We are more looking into the regenerative state at the pollen grain. So as I said, because that's happened, what happened is that during the high temperature, we see in the field, many colleagues who are working on chickpea as well notice that when there is high temperature during flowering, a lot of flower abortion happen. So we like to see that how that happened. So, and then clearly showing there's changes in the morphology, the structure of the pollen grain, and then in the fertilization. So that's what we are trying to aim. So, but probably that can be broadened how this adaptation occur during the process, during the evolution. So that's the next question we would like to explore.
Okay.
Thank you. Thank you,
Thanks.
Nelson.
Thanks, Bunyam. Okay, I don't see any other questions, so let me just check the chat very quickly, but nobody's writing. Okay, so thank again, Bunyam, for this nice, a very interesting presentation. It's a very interesting topic that we are bound to face a lot more in the future.
Yeah.
I thank again all the everybody that's in the audience. Our webinars will come back next autumn after, hopefully, the heat starts to give us a little bit of breathing room. Thanks again. Thanks to everybody. Thanks especially to those that were at our conference in Dubrovnik. And we'll see you again next season. Bye.
Bye. Thank you.
Thank you so much.
Thank you, everybody.
Bye. Thank you.